This video we are going to discuss about oxygen hemoglobin dissociation curve. This is very important topic from exam point of view, and it has many important concepts. So oxygen hemoglobin dissociation curve is basically a graph here. It is a graph which is plotted between percentage saturation of hemoglobin and myoglobin and partial pressure of oxygen. So when we plot the graph of hemoglobin, then it comes about like this. It is like this, or it is you can call it S-shaped graph. While if we plot the same graph for myoglobin, then the graph for myoglobin comes like this. So the graph of hemoglobin here is sigmoidal in shape. The graph of hemoglobin is sigmoidal in shape or S shape, while graph for this is for hemoglobin Hb and this is for myoglobin, and for myoglobin the graph is hyperbolic in shape. So what does this change in shape signifies? This is the percentage saturation here, and here comes about the 50% saturation here. Okay, and if we count here, then 50% saturation of myoglobin is achieved at very low partial pressure of oxygen, and it is usually at about 1 millimeter of Hg. At 1 millimeter pressure of Hg, 1 millimeter pressure of the partial pressure of oxygen, the myoglobin is 50% saturated, while 50% saturation of hemoglobin occurs late, and it is about at the partial pressure of 26 millimeter of hemoglobin, and here is 40 millimeter Hg, then here is 80 millimeter Hg, and here is then 120 millimeter Hg, and so on. So myoglobin is saturated early or at very low partial pressure of oxygen, while hemoglobin is saturated late or very high partial pressure of oxygen. So what does it show? It shows that myoglobin has high affinity for oxygen, while hemoglobin has less affinity for oxygen. So hemoglobin has less affinity. It means that it does not bind easily to oxygen, while myoglobin binds much easily to hemoglobin, and the thing that binds much easily releases with difficulty. So myoglobin binds easily to oxygen. It binds easily to oxygen, while releases oxygen with difficulty because it has high affinity for oxygen. Myoglobin has a high affinity to bind oxygen, so it binds easily while releases oxygen with difficulty, while hemoglobin has less affinity comparative to myoglobin, so it binds with some difficulty. However, it releases oxygen easily as compared to myoglobin. So what does it signify? It signifies that at 1 millimeter, even at the partial pressure low as 1 millimeter of Hg, myoglobin is about 50% saturated. However, at the pressure of 1 millimeter Hg, hemoglobin is very less saturated. It means that at this partial pressure of oxygen, hemoglobin has released nearly all of its oxygen content. So hemoglobin, what does it show? They show that at this partial pressure range, hemoglobin has tendency to lose oxygen. However, the myoglobin does not have tendency to lose oxygen at this partial pressure. Instead, at this partial pressure, myoglobin is saturated. So myoglobin releases oxygen at very low partial pressure of oxygen. So myoglobin is basically transport protein in skeletal muscle. Due to extreme work, the partial pressure of oxygen drops very low. So myoglobin proves a good oxygen channel in the skeletal muscle at very low partial pressure of oxygen. However, for the rest body tissues, it is present in skeletal muscle and cardiac muscle. However, for the rest of the body tissues, hemoglobin is a good transporter because hemoglobin can release oxygen at a varied range of partial pressure of oxygen, while myoglobin only releases oxygen at very low partial pressure of oxygen. Okay, for example, if a tissue has partial pressure of 20 millimeter Hg and it needs oxygen, then myoglobin will not be able to release oxygen there. However, at 20 mm of Hg, hemoglobin can release oxygen. So hemoglobin is a good transporter of oxygen for the different tissues of body, while myoglobin is good. Myoglobin is the storage of form of oxygen to release oxygen at very low pressure in the skeletal muscle and cardiac muscles. So it was a comparison of myoglobin and hemoglobin. So what does this graph here signifies? The curve for hemoglobin is sigmoidal in shape. This curve shows that the hemoglobin can release oxygen at the varied range of partial pressures, while myoglobin has higher affinity. Its curve is hyperbolic in shape, and it can only release oxygen at very low partial pressure as compared to hemoglobin. So myoglobin is not a very good transporter of oxygen to tissues, while hemoglobin is a good transporter. So another thing here is about allosteric regulators. So these allostatic regulators are different compounds which regulate the binding of oxygen to the hemoglobin. These allostatic regulators only affect hemoglobin. They does not affect the binding of oxygen to myoglobin. So next one we will discuss the allostatic regulator with respect to hemoglobin only. We will not discuss with respect to myoglobin. Okay. So another thing, myoglobin only bonds one molecule of oxygen due to one heme group, while hemoglobin binds 4 molecules of oxygen due to presence of 4 heme groups. Okay. So next, another thing we should be knowing that there is two forms of hemoglobin. One is T form and one is R form. T form is taut form and R form is relaxed form, and it is the relaxed form is the oxyhemoglobin, or it is bound to oxygen, while taut form is deoxyhemoglobin. So what does it mean? Relaxed form has high affinity for oxygen, it is bound to oxygen, and it has high affinity for oxygen, while it has low affinity for oxygen. These things we have discussed previously in previous lecture. Moreover, the binding of oxygen with hemoglobin is sequential. For example, one O2 molecule facilitates, when one molecule binds to hemoglobin, it facilitates the binding of other molecules due to breakage of some bonds between hemoglobin dimers. So one molecule facilitates the binding of other molecule or increases the affinity for other molecules. Okay, so one molecule binds, it creates easiness for second molecule creates easiness for third molecule, and third molecule creates easiness for the fourth molecule. So affinity of hemoglobin for oxygen increases as more and more molecule bind with hemoglobin. However, when one molecule is unloaded from hemoglobin, then the ability of hemoglobin to bind oxygen decreases, or affinity of hemoglobin decreases. Then there is easy release of second molecule, and when second molecule release, affinity further decreases. So third molecule is released more easily, and then fourth molecule released more easily, and so on. So as binding of oxygen with hemoglobin increases the affinity, the unbinding of oxygen with hemoglobin decreases the affinity. So when one molecule binds, it increases the affinity and helps the binding of other molecules, and one molecule is removed, it also helps the removal of other molecules. So this thing makes the hemoglobin a good transporter of oxygen from lungs to the tissues. In lungs, what happens? Supposedly here are the lungs. Here in lungs, oxygen is abundant. So here there is easy loading of oxygen to hemoglobin. One molecule binds, then second, then third, and fourth. And when it moves to tissues, in tissues there is low oxygen. So one molecule is released from hemoglobin, and this loss of one molecule facilitates the removal of other molecules as well. So here hemoglobin unloads easily. Here it loads easily, and here it unloads easily, and this phenomena is called heme-heme interaction. This phenomena of hemoglobin which facilitates the loading of hemoglobin in lungs and unloading in tissues is called heme-heme interactions, and it makes hemoglobin a very good transporter of oxygen from lungs to the tissues. Okay, so these are the heme-heme interaction which facilitates the binding of oxygen with hemoglobin in lungs and facilitate the removal in the tissues. Okay, so this is how the hemoglobin is transporting oxygen. Next we should be knowing here the Bohr effect. Another thing we should be knowing here is the Bohr effect. Bohr's effect. So what is Bohr effect? Uh, we know that in tissues there is metabolism going on which produces lactic acid, which produces carbon dioxide, which is converted into H2CO3 or carbonic acid. So both these acids produced, what they do, they release H positive and decrease the pH. So this decrease in pH, what it does, this decrease in pH favors the unloading of hemoglobin. It favors the unloading of hemoglobin in the tissues. So when pH decreases, decrease in pH decreases the affinity of hemoglobin for oxygen. So when pH decreases, affinity of hemoglobin for oxygen decreases, and when affinity of hemoglobin for oxygen decreases, what happens? There is easy unloading of hemoglobin. So this decrease in pH increases decreases the affinity and shows the easy unloading of hemoglobin in the tissues, and this phenomena occurs in tissues. In tissues, due to metabolism, acids are produced which decrease the pH, decrease the affinity of hemoglobin, and easy unloading of the hemoglobin occurs. So there is release of oxygen from hemoglobin to the tissues, and this phenomena occurs in tissues. So what is the effect of this on the oxygen hemoglobin dissociation curve? Supposedly here what the curve here, and in tissues there is decrease in pH and decrease in affinity. It means that supposedly here it was 50% saturated and 26 mm Hg. No, affinity of hemoglobin for oxygen has decreased due to decrease in pH. No, it means that hemoglobin is binding less oxygen and releasing more oxygen. So this curve shifts forward towards right side. Now this curve shift towards the right side, or right shift occurs, because now this 50% saturation will be occur at higher pressures. At lower pressure it has released oxygen. So decrease in pH shifts this curve towards right side because affinity of hemoglobin for oxygen has decreased due to presence of H positive ions. And how this phenomena occurs, or what is this mechanism? This mechanism is because the histidine residue present in hemoglobin, when they are in deoxy state, they pick up the hydrogen atoms, and form HBH. Here is HbO2 plus H. What happens? This H causes the unloading of oxygen is released, then this H binds to the histidine residue and form HBH in the tissue. So HBH is formed, and when it moves to the lungs, what happens? In lungs there is high pH. And this high pH, what it causes? High pH means there is basic environment. So this basic environment causes the release of hydrogen from hemoglobin, and oxygen moves to the hemoglobin. So what is known? Let's summarize the Bohr effect. In tissues, acids are produced which decrease the pH. This decrease in pH decreases the affinity of hemoglobin for oxygen, so hemoglobin releases oxygen to the tissues. In turn, hemoglobin binds hydrogen and HBH is formed. This HBH goes to lungs. In lungs, what happens? In lungs, pH is higher. This higher pH causes release of hydrogen from hemoglobin, and in turn oxygen binds to the hemoglobin. So this Bohr effect in turn facilitates the transport of oxygen by hemoglobin, and it causes a right shift in the oxygen hemoglobin dissociation curve. Okay, so the same phenomena occurs due to presence of carbon dioxide as well. In tissues, carbon dioxide is produced. Carbon dioxide is transported in the form bicarbonate ion as well. However, some carbon dioxide also binds with hemoglobin. Here is Hb, and hemoglobin has an H2 group as well. So carbon dioxide binds here and forms carbamate ion, NHCOO negative, okay, plus H. So in this form, hemoglobin also transport carbon dioxide, and in the lungs this carbon dioxide dissociates and released out, and this carbon dioxide present in the tissues also stabilizes the T form. It stabilizes T form of the hemoglobin or tau form. T form is the deoxygenated form. So this as it is facilitating the stabilization of tau form, it means that this carbon dioxide in tissues also causes the release of oxygen. Okay, so this carbon dioxide also facilitates the release of oxygen in the tissues and decreases the affinity of hemoglobin for the oxygen. Next, we move on towards an important regulator of the hemoglobin oxygen transport, which is 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate. It is an intermediate produced from the glycolysis. In glycolysis, glucose is converted into 1,3-bisphosphoglycerate, and then 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, and 1,3-bisphosphoglycerate is sometime converted into 2,3-bisphosphoglycerate, which is an intermediate, and this intermediate 2,3-bisphosphoglycerate is an important regulator of the oxygen transport by hemoglobin. What does it do? It binds to the hemoglobin. And decreases the affinity of hemoglobin for oxygen. So what does it do? It shifts the curves towards right side. It causes right shift of the curve. And normally it is present in RBC in some amount. And facilitates the transport of oxygen. However, if it is not present, there will be left shift of curve. So absence of 2,3-bisphosphoglycerate increases affinity and reduces the ability of hemoglobin to transport oxygen. So this decrease in ability of hemoglobin due to loss of 2,3-bisphosphoglycerate is observed in the stored blood. Stored blood has decreased 2,3-bisphosphoglycerate, and when 2,3-bisphosphoglycerate in stored blood is decreased, then this stored blood can prove a oxygen trap. It can prove oxygen trap because this blood cannot release oxygen, because 2,3-bisphosphoglycerate is necessary to decrease affinity of hemoglobin for oxygen, and when affinity will be decreased, then there will be easy release of oxygen from hemoglobin. So let me summarize. 2,3-bisphosphoglycerate is an intermediate of glycolysis, and here is the oxygen hemoglobin dissociation curve. 2,3-bisphosphoglycerate causes a shift towards the right side and decreases the affinity of hemoglobin and causes easy removal of oxygen from hemoglobin to tissues. It causes easy unloading of oxygen to tissues. However, if it is absent, then there is shift towards left side. The affinity is increased, or there is difficult removal of oxygen. So there is difficult unloading of oxygen to tissues, and this decrease in 2,3-bisphosphoglycerate is observed in the stored blood. So stored blood can prove an oxygen trap. So different compounds are added to the stored blood, and different conditions are maintained to maintain the 2,3-bisphosphoglycerate in the stored blood. And 2,3-bisphosphoglycerate is increased in response to the hypoxia, high altitude, as well as anemia, to facilitate the more and more oxygen delivery to the tissues and prevent this oxygen trap. Next, we should be knowing the role of this is the normal oxygen hemoglobin dissociation curve. Now we should be knowing the role of carbon monoxide. What does it do? Carbon monoxide binds to it and increases the affinity of oxygen from hemoglobin. It increases the affinity of hemoglobin. So there is left shift here. As affinity of hemoglobin for oxygen increases in response to carbon monoxide binding. So affinity has increased, so it also causes hemoglobin to become an oxygen trap. So carbon monoxide binding to hemoglobin causes hemoglobin to convert into oxygen trap, because affinity of hemoglobin for oxygen has increased. So increased affinity means there is difficult unloading. However, this condition can be reverted by 100% oxygen therapy. It means that if patient breathes in 100% oxygen, which is called hyperbaric therapy, it will remove carbon monoxide from hemoglobin and restore its ability to transport the oxygen. Okay, so it is the effect of carbon monoxide which decreases the ability of hemoglobin to transport or to deliver the oxygen to the tissues. So it was about the oxygen hemoglobin dissociation curve and the effect of different allostatic regulators on the oxygen hemoglobin dissociation curve. Thank you.